Good morning and welcome to our seminar today. It's sorry. It's a pleasure to have David Marshall talking to us today. David is currently working at the Scotland Rural College in Scotland, and he retired from the James Hutton Institute after decades of work focusing on plant genomics and understanding patterns of genetic diversity in plants. His major focus has been on developing software tools to analyze SNP variation in crop plants, genetic resources, and breeding applications. Well known for his work in developing visualization tools, and his talk today relates to that. Seeing is believing, data visualization, genetics, and breeding. Have worked together with David for a few years in YAMS. I tried to include photos of the crops he has worked or helped through his career. I imagine I missed some of them, David, but welcome to the seminar, and thank you for taking on the challenge. I'm going to stop sharing now and give the floor to you, David. Thank you.
Thank you. Thank you very much, Carlos.
It's good.
Okay.
We can see you, sure. Yeah, we can see it.
Okay. So my title is Seeing is Believing, Data Visualization in Genetics and Breeding. Really, what makes good visualization? I've stuck up here a picture of a book called Visualization, which is something which I think is well worth a read for people, by David McCandless. And it basically says information is beautiful. And this is something I felt for many years in my career. But if we're thinking about what makes a good visualization, there are a number of components to it. There's the actual information, the data that goes into it. There's the story that we want to tell. There's the goal, what's the function, what's the whole point of doing the visualization? And then there's the how do we actually do the visualization? What's the metaphor that's being used to visualize information? And all these are important considerations if you're going to visualize data and make decisions based on the visualization. So I want to start off by saying there are a number of major genetics drivers. These have come from advances in sequencing, genotyping, and phenotyping technologies. And one of the consequences of this is that the data volumes have grown enormously. And there are new quantitative approaches to plant breeding, such as genomic selection. And plant breeding is essentially a numbers game. So we have a large number of entities that we need to deal with and make decisions on. Another really important thing is that key decisions are often needed to be made in a very short time window. And visualization can be really useful in terms of helping that. And they often require integrating multiple components of information, so multiple pieces of information. So if we're thinking about the technologies behind it, I think one important thing to say is that if you're handling a lot of data, we've moved beyond Excel. There are nice graphic libraries, for example, for R, for Python, for Java, and other languages. An example of that is the ggplot family of graphical applications. It's also possible to write your own bespoke applications. There's an increasing value in using various web page technologies. But some of the considerations are the scale at which you want to do something, and performance and responsiveness is very important. People like to be able to things to happen quickly and dynamically. There's also an issue in terms of the choices of colors that you use as well. There are issues in terms of color blindness. I'm not going to make any consideration of that. But if you are colorblind, you may have to think about what palettes you use, what combinations of colors. There's also an issue in terms of that there are only so many colors you can discriminate between. And that's often an important consideration when you're trying to make decisions based on visualization aspects. And also a final key element here are data standards. And I'll say a little bit more about data standards towards the end of the talk. So I'm going to go right into talking about a particular application that we developed what is now many years ago. The application is called Flapjack. We made a decision very early on that in choosing names for applications that if you tried to do something that actually talked about their function, it was often quite tortuous and quite difficult to differentiate yourself from other things. So we abandoned that completely. And for a while, we used food names as a way of describing our applications. So Flapjack is essentially what you can call a graphical genotyping application. It's a way of visualizing genotype data, but in the context of map information. You can also use QTL information alongside it. You can integrate it with trait information as well. And what we have here is the basic view that you get if you load data in. You have this colored patch here, which is the SNP data. You have essentially SNPs in either in map or genome order going along here. You have different lines going down here. You have a little overview window because we deal with very large data sets. So once you get to a point of having thousands of SNPs and potentially thousands of lines, you need to be able to migrate around the data set. The data can be brought in through a number of routes. It can come in through flat files. It can come in from a database. It can come in through a repository, and with very large data sets, it needs to come in from a binary repository. And it's also worth stressing that we're talking about a visualization application. It has some analysis capability, but it's really about bringing pre-analyzed data together. It scales very well to large data sets. And if you really are interested, it plays Minesweeper as well in the background by an appropriate combination of keys. Okay, let's look at an example data set here in Flapjack, the application. So this is a UK barley data set here. It's based on 50,000 SNP markers, a genotyping chip. We have an overview here of part of one of the chromosomes. If you look in the overview window here, you can see there's a little highlighted area, and that's what you're seeing on the screen, the main screen here. So we have, this is chromosome 1H in barley. This is a window which is visualizing the trait data here. And this is the main SNP window here. And this is genotype data, and it's visualized with a different color for each of the nucleotides for the SNP. Now we can sort the data here, and if you look here, this trait heat map here has changed, and these are winter barley lines in red and spring barley lines in green. And if you look already, you can see that there's a difference between the two sorts of lines just by seeing the differences in patterns start to come out. We can enhance this by, rather than visualizing the actual nucleotide, we can visualize by choosing an individual line and visualize by similarity to a line. So if we choose one of the spring lines, we can see that the spring lines look very similar, and they're largely green. There's a few bits of variation within the spring, but the winter lines look very different, and you can see by the occurrences of red here. Again, apologies to the colorblind, but you can change the coloring if you require and use a different combination. So that's just a basic idea in terms of functionality. Let's look at something a little bit different. We'll use a smaller data set now. This is again barley data. It's a doubled haploid population of barley. It's based on what is quite now an old-fashioned genotyping chip with just 1,500 SNP markers on it. And the parents for this were two commercial barley varieties. And so this is part of chromosome 7H, or in fact, it's essentially most of chromosome 7H from this smaller data set. And what we have is a whole series of doubled haploid lines down here and the two parental lines as well. If you look in here, there's regions here which have a lot of variation. There's regions which look like they've got very little variation, more regions here with variation. And this is because we've taken no consideration as to whether the loci here are polymorphic or not. And these are two relatively closely related lines. So there are regions of identity by descent. So what sort of things can we do? Well, we can go into the application. We can identify, if you look here at chromosome 7H, there are 158 SNP markers here available to us. We can choose a color scheme here that by choosing a color scheme, the original one was a nucleotide model, but this gives you an idea of the range of color schemes. So we can choose an anchor line. If we choose an anchor line, which is one of the parental lines, when we do that, you have a whole series of lines for this region here on this chromosome. They're almost identical to the parental line. And clearly we have blocks of polymorphism along the chromosome. We can then do sorting by similarity as well as by coloring by similarity. And if we just select a small region here, perhaps this is a region where there's a QTL, or a region that we're interested in for a gene that we're interested in, we can see there's probably quite restricted variation in this region here. And if we sort by similarity here, we found that there's lots of the lines look like one parent, lots of the lines look like the other parent, and there's a small number of recombinants in the middle here. So we can quite easily and quite nicely start to dissect out the variation and look for patterns at a local level as well as at a wider level. Okay, so that was with a small data set. We can also work with the larger data sets. And this is an example here from rice. Some time ago now, there was a project, an international project, which sequenced 3,000 genomes of rice. And the data from that is available on the SNP-seq database. So this is sequence data from 3,000 different rice genomes. And you can download and extract the data in various different ways. And one of the ways you can extract the data is in a format which you can put it quite nicely into our application, Flapjack here. You can choose a particular chromosome, you can choose a region in a chromosome, or you can choose an individual locus. So we can extract the data from a chromosome region or for one or more genes and format the output for Flapjack. So this is the data that we've downloaded from this database, and we've loaded it into Flapjack. And the regions here we've chosen is a region I have some interest in, which has two nitrate transporters. And on one side of there is a gene here, a nitrate transporter, a retroelement between the genes, the other gene, and another the other nitrate transporter, and another flanking gene. And we've got a stack here of 3,000 lines. So we're only looking at, in this case, at the top of the stack here. We can highlight just the SNP data from an individual gene. So if we unhighlight all of the genes to start with, we choose this gene here, and we can do it by clicking on the gene. So we're using what was a QTL in our previous example is now acting as a gene. We can just click on that and identify the variation under that gene. We can go to the next one. So this is one of the transporters. We can go to the retro element, the next transporter, and the final flanking gene. So, so what else can we do? Well, we can look at all of the haplotypes in this region quite nicely. So I've broken this stack because there are 3,000 different rice varieties here. I've broken the stack into 4, but you can imagine this is on top of this, which is on top of this, which is on top of this. And we see here, on this part here, we have a range of varieties which are all identical. There's no SNP variation at all. There's some here have a single SNP variant. We start to get a region with more SNP variant. And by the time we get to some of the ones which are very different from the one we started with, we find there's actually a presence or absence of this flanking gene here, as well as presence or absence of the retro element. So we can handle really quite large data sets, and we can understand a lot about the patterns of variation in them using this type of visualization approach. Okay, let's go and look at something a little different. So another way you can look at a large number of data lines is by using this SNP data and using either a principal coordinate analysis or a principal component analysis. And by doing that, you end up with a multi-dimensional view of the data. So what we wanted to do was to develop an application which enabled us to do something a bit more dynamic than just a simple plot. So this application is called Curly Whirly. This is a chocolate bar in the UK, which is where we chose the name for this. And this is the application, just looking at a screenshot from the application. So we have a really quite conventional 3-dimensional plot here. You can spin the plot round, so you can look at it from different aspects. But the other nice feature is that you can color it by various categories. This is a barley data set here, and you can color it by, for example, seasonal type in barley. So you can, the light blue ones are spring, the purple ones are winter. There are green ones which are alternatives, so these are varieties you can use as either sort. And there's some which the data didn't say what they were. So you can use this to play around with the data, to look at subcategories of the data. So this sort of thing, and let's look at some examples. So this is a European maize landrace data set. And here we've just colored it by the different groupings. So these are colored by the different types of maize. And again, they separate out quite nicely. You can spin it around, you can query, you can put the cursor over it and find out what any individual one is. Again, just in case anybody in North America doesn't know what maize is, I've got some nice examples of hybrid maize or inbred maize growing at Simmet. And just to show that we are not ignorant of maize either, this is maize growing in Scotland at 56 degrees north, forage maize. Okay, and again, you can do fun things with it. You can run a little movie from it as well if you want to, but just to emphasize and show you what it looks like in terms of the ability to manipulate in 3 dimensions. Okay. Okay. So let's look at other things you can do with it. So one of the questions that people had was that if I have a line that I'm interested in, but perhaps I don't have enough seed of that line, can I find something close to it? So again, we've set it up here so you can choose a target line, and then you can develop as it were, a window going out from that target line and highlight ones which are similar to that in 3-dimensional space. And then you can use that as a basis of making choices. I want to look at some other examples using this application. I'm basing these examples on data sets that come from the Seeds of Discovery project at CIMMYT in Mexico. This project was funded by the Mexican government and a little bit of funding from the BBSRC in the UK as well. And it used the DART-seq variant of GBS technologies to genotype a significant proportion of both the CIMMYT maize gene bank and much of the wheat germplasm that came from the CIMMYT and Acarda gene banks. So it's basically a large number of lines and a large number of markers. I just want to acknowledge the important role that Sarah Hearn, Carolina Sassolini, and Mario played in generating these data sets. Okay, so this is a 20,000 line tetraploid wheat data set. And we've just colored it by a couple of different categories. So so we've colored it by the gene bank of origin. So we've colored it by the CIMMYT ones are red and the Acarda ones are blue. So you can see where the different lines come from. But you can use the same basic layout and color them in different ways. We can color them by country of origin, and you can see there's this almost lens down here, and these are Ethiopian ones, which are quite different from the rest. Or you can do a cluster analysis, and you can color them by the different components of the cluster. So these are just colored in this case by the different cluster groupings. Okay, so you can do a similar thing with hexaploid wheat lines, and this is a 60,000 line data set. Again, these DART-seq markers it's based on. Again, the CIMMYT lines in this set are colored red. The Acarda ones are colored blue. We can switch off the Acarda ones and show the CIMMYT ones. In the plot, we can do it the other way around and switch off the CIMMYT ones. And we can, again, cluster by different cluster groups and color by those to show the major groupings in the data set. Okay, well, this is a slightly different set. So this is based on wheat wild relatives in the collections, again, that come from CIMMYT and Acarda. And again, you can see that the different gene banks actually hold a different sorts of collections of wheat wild relatives. When they're assigned to species based on the passport data, this comes from the passport data. They color up, although they don't fit into all the nice, neat categories. So there's not a nice blocking of the passport data in terms of where they come on the clustering. And if you carry out an actual clustering based on the market data, they cluster quite nicely. And my suspicion is very much that quite a lot of the passport data is not saying exactly what these different wheat lines are. Okay. So that's a couple of different applications. Now let's move on to one other major visualization. So again, we quite interested, or became quite interested in starting to visualize pedigrees. One of the reasons for this, for many of the inbred cereals we work with, we have good pedigree information. In formal terms, pedigrees can be described as a directed acyclic graphs, known in short term as DAGs. And there are a number of tools and algorithms which can be used to draw and manipulate DAGs. Okay, so what do pedigrees look like when you lay them out using these sorts of tools? So this was an example that we were interested in using pedigree data. And this is just an interesting thing showing the French influence on UK wheat breeding in the 20th century. In the early 20th century, UK wheats were dominated by French varieties, and there was a strategic decision to start up a public-funded UK wheat breeding program. to compete with the French varieties. And these were developed at the Plant Breeding Institute in Cambridge. And all these lines had a prefix on their names called with Maris. And this is just a layout using a standard DAG tool called GraphViz. And you can see here this line here, Capelle d'Esprit, which was one of the very successful French lines, has been a major contributor even to many of the derived UK varieties, which are coloured in pink. So again, it just gives you a nice overview of a breeding program and its relationships. Again, you can do these things on a much larger scale, although they become a bit harder to deal with them in terms of static overviews. This is the pedigree information for UK elite barley cultivars. The ones which are pink are spring barley. The ones which are grey are winter barley lines. The other ones are ones which can be used as either. And we've changed the size of the actual variety according to how much it's contributed as a parent. And you can see one or two lines have contributed very highly as parents. So they've been major parents in the overall programs. Okay, so having started off realizing we can do this, we thought we'd then translate this into an application, which was a bit more dynamic. So this application was developed by colleague Paul Shaw in our group at James Hutton to give much more interactive control over pedigree visualization. It uses simple data formats and coloring, and you can color it by categorical information. You can color it by genotype or haplotype. It consists of a major overview window for the actual pedigree. So we're looking at this little bit in the overall pedigree, and you can extract sub-pedigrees out of it. And you can also use trait data or other categorical information to color the different elements in the pedigree. So again, let's look at some pedigree examples here. So this is a winter wheat pedigree. And winter wheats are the most widely grown wheats in the UK. In this case, we've coloured by breeding company. And again, it just shows the relationships between the different breeding companies, because the different companies are able to use other companies' material as parents. The material has lots of cross links between it. If you also look here, there's one individual variety that looks like it's been used very widely as a parent. Again, we can change the size of that to show that up much more explicitly. This is a variety called Robicus. We can extract that as a sort of sub-pedigree, which shows that Robicus is a parent or grandparent of a large number of different varieties. And we can do a similar thing with another example here. This is a barley variety. There's one variety here called Maris Otter. It has a similar prefix to the earlier wheat example, which shows that it came out of the Plant Breeding Institute in Cambridge. But this variety has been very, very successful, in particular in terms of varieties of barley that have been used as for beer or for whiskey in particular, and it's still grown in specialist areas. In this case, the spring barley varieties are highlighted in purple, and winter ones in this red color. Okay. You can do this on a very large scale if you want to, just to show its scales in principle, although it's quite difficult showing it all at once. You really need to view small parts of the pedigree. This is an attempt that's been made to integrate the data from really all of the UK wheat breeding and going back to the earlier parents of it. but you can link it very, very nicely by pedigree. Okay, let's just finish off here by starting to think about some more general considerations. Basically, data standards are key to data and data integration. And this is one thing which has become really important as data volumes have grown. But also we want to be able to integrate data from a number of different sources. And a key important aspect is data quality and rapid data pre-processing. So if you're going to make decisions based on a lot of data and make quick decisions, you need to pre-process your data quickly, and you need to really look quite hard at data quality. And quality checks are often aided by visualization, and I emphasize that this is best done at the point of data capture. Data integration between data capture, storing it, analysis, visualization, and decision support requires what we call data interfaces. And data interfaces are really important. Basically, we need ways of pooling data or different data types together. A major effort has been made in order to standardize this, an effort called BRAPI, which is the Breeding API, or Application Programming Interface. This was an initiative that was started off by the Gates Foundation and has a large number of partners, both in public programs and even commercial companies around the world. So it presents, for example, genotype data in a standard way, or data about line information in a standard way, or trait information in a standard way. So your application knows exactly what it's getting. Just to summarize a little bit here in terms of where visualization can help, it can really help at really almost every stage in the breeding program, and a pre-breeding stage. In terms of quality control, I've not talked about this, but it can be really useful for being able to quickly scan, for example, marker data on clonal lines to identify abnormalities or contaminations. It can be very useful for integrating marker and trait data. In terms of making parental choice, it can be really useful when you bring together trait data and marker data for a large number of lines. You might often, for example, have a line which looks very good, but you don't have enough seed of it. So you might want to have another very similar line, and you want to check just what that line is and how it might perform. And this can be really crucial at every stage up to product advancement meetings, where you're making choices about make lines to take forward for crossing, or lines that you actually want to commercialize. Just a shout out here to some work from colleagues, which has been at the pre-breeding stage, which is looking at splitting your genomic selection approach into being able to separate the information from the markers from the advanced lines from the more exotic lines, so that you ensure that you don't lose key elements from the exotic lines in the different cycles of genomic selection. Okay, so just to say that integrated solutions are really important. It's quite an effort to be able to do this. For more sort of commercial context, we've been involved in a joint project with company VSNI, with Biometris in Wageningen, under this heading called H3 Analytics, when we're trying to provide an integrated components that allow you to go from gathering data in the field, involving and looking at design information, phenotype data, pedigree information, data quality control, making predictions, and final decision support, and integrating different components. I want to say, just as I finish off here, a little bit of a rant about funding software. The development, maintenance, and tailoring of software tools is of increasing importance for plant breeding and genetics, and it's true for many other aspects and areas of genetics. It's often possible to get funding for the initial development of software components, but the maintenance and incremental development of them is much harder to fund. And this is a major issue because in an area of data-intensive science, it's important that software is considered like bricks and mortars or field equipment. You need this stuff to be able to go. You need to not just be able to hack something together, you need it long term, and you need it to be able to change in terms of as computers change and evolve, as data changes and evolve, you need to be able to maintain and improve your software. I thought this was worthwhile showing as well. One of the things that we've done just to help, as it were, persuade our funders that our software is good and people are interested in using it. We keep a map which just shows roughly where in the world people have been downloading and using our software. So this is just an example here of Flapjack and an idea as to where in the world people have been using it. For a while, we found each new point was exotic for a while, but they lose their novelty after a while. Okay, just want to finish off by some acknowledgements. The Flapjack application was the major developer of that was Ian Mill, colleague Gordon Stephen put a lot of effort into it as well, and Bill Thomas, one of the barley geneticists, made a great deal of contribution towards it. Helium, the pedigree application, is due largely to Paul Shaw, and Curly Wurley is due to Gordon Stephen, Ian, and Sarah Hearn. But what's been really important is the input from colleagues at CIMMYT from a whole range of different, largely UK, but also European plant breeding companies, and many colleagues at both James Hutton Institute and SRUC. I also put a thanks in here to William Playfair. I think it's also worthwhile highlighting his role. William Playfair was the person who really invented statistical graphics, and it comes from not far away from where I was sitting, but a long time ago. Our funding has come from the BBSRC, from the Scottish Government, a surprising amount of funding from the Mexican government through SAGARPA and the Mass Agro program, and also the Gates Foundation. All of the software which I've talked about today is freely available and can be downloaded from the website that I've put a link to here. And I just ask that if anybody uses the software, that they acknowledge it. If anybody would like to contribute to the development of it in financial terms or input, I'm sure getting in touch with colleagues at James Hutton would be gratefully appreciated. Okay, thank you very much.
Thank you, David. It's been great, great talk, and we already have some comments and questions in the chat. Please post questions and comments in the chat. Ben is saying, looking forward to using Helium for my own projects, and thanks for the presentations. Sandeep is asking, are there various platforms, applications presenting this talk open access? And you referred to that at the end, isn't it?
Yes, they're all open access. And again, this is something which we've felt is really important. But I will say that we've tried to constrain input and development, so they've all been developed through one group of people rather than being very large number of different contributors. But they actually, through initiatives such as BRAPI, we also link into to many other more open projects.
Can those tools be used to analyze RNA-seq data for microorganisms?
No, these are not really about RNA-seq data. But if you look at the software pages on the Hutton website. There is an application there called 3D-Seq, which is available for RNA-seq data analysis. We also have a different application called Tablet, which can be used for really visualizing sequence data. It works really nicely with Illumina sequence data or nanopore data as well. And has been really useful for developing analysis methodologies where we can visualize really how well the sequencing is operating, and we can find odd things in terms of nucleotide calling and sequencing.
Flavio's asking, when is it wise to go for paid software for a plant breeding program?
That's a difficult question. One of the problems is that much of the, as it were, commercial programs are really quite expensive, and it really depends on where the money comes from for your software. There are some things which are really nice, which are out there, but they can also be quite closed as well as you. So they may not do exactly what you want as well. So, but a lot of it depends on scale. It's bespoke software is expensive, and there's no getting around that. So we've been lucky that we've been managed to get, as it were, academic funding and put that into our software development, and that means that the software we provide is free. But there are major issues in terms of the costs of much of the commercial software that's out there. Although some of it it depends on where in the world you're coming from as well. So there are some opportunities, for example, in developing countries where you can get massive discounts or free access to software. And there are some initiatives through the Gates Foundation, through CGIAR, where you can get access to free software. But it's not just the issue about buying the software; it's supporting the software as well, which can be a major issue, because again, the more complex it is, the more you can easy it is to make mistakes and do the wrong analysis.
So you talk at the end of your presentation about these interfaces like RAPI. What are the opportunities into the future in terms of developing visualization tools? What are the gaps, and what is the next frontier?
I mean, we've not done a lot in terms of really, I suppose, fully integrating visualizing the traits and the marker data together. I think there's quite a lot of really quite sophisticated things that you can do that. So we have a lot of concepts now that which work on a sort of static level, but don't work really dynamically. But there's a really nice things where you can do sort of what if experiments, where you can use as it were simulation software along with the marker data or trait data that you have, which can help you make decisions in terms of the design of your breeding programs and the likely outcomes. There's a really nice suite of breeding program simulation programs called AlphaSim that comes from the Roslin Institute in Scotland, and that has really nice capabilities in terms of simulating breeding programs.
Thank you, David. Also, as a reminder to people, we're going to post the recording of this presentation, and maybe if we can get the PDF version of your presentation, David, because the website is there, we can put it together, okay? Yep. Thank you so much. If any other questions, please post it in the chat. I'm gonna, in the meantime, share our announcement for next seminar next week. It will be the last seminar of the series at the same time next Thursday. Some art sheets from the Leibniz Institute in Germany. DSMZ. We'll talk about virus resistance, breeding virus resistance for virus resistance in cassava. An interesting piece of work that this institution in Germany has been done in collaboration with international centers and with national programs in Africa. So I invite you, all of you, to participate. Thanks, Brandon and Thiago, for your help. Thank you so much, David, for having you speak to us. It's a great pleasure, and we really enjoy your presentation. Okay, everybody have a nice rest of the week and a good weekend, and hope to see you next Thursday for our last seminar of this series. Have a good time, David.
Okay, cheers. Thanks again. Again, I'm happy to respond to emails if people have specific questions.
Okay, so there's one more question here. Is that possible to get additional data formats like AppMap? From your website? I don't know what's really CETA is asking.
Yes, but again, it's a relatively simple scripting issue to make that happen. Yeah.
All right. Thank you so much, and see you next week.
Cheers.